SUSHI stands for super resolution shadow imaging, and it refers to the approach of imaging with an inverted contrast. The idea is not to label the structures, the cellular structures from the inside, but rather label them from the outside, which blocks you from obviously seeing anything on the inside, because you have no contrast inside of the cells. But if you're interested only in the morphology, of the anatomy of the system, of the tissue, then this is, I would say, a much superior strategy, because it saves you this very invasive step of putting a dye into a cell, either mechanically or by genetic engineering. It's very, very easy to be applied, and it's provides you with the same contrast in theory. Yeah? Even better, because you're labeling only a minority of the space. Yeah? It's sort of more economic in the sense of giving background noise and so on. It's better than labeling the majority of the space. Yeah? You label a minority, yet you get as a contrast that is as high or even higher. And then it has the benefit of seeing not just one cell or a few cells that you're labeling, you happen to label, but you see the contours. Through the contours, you see all of the cells, and you're not, with sufficient resolution, this is where super resolution came in, you're not missing any detail, any morphological detail of the brain tissue, and you can sort of get a comprehensive view very easily.
And how do you label your cells?
Yeah, so we label the tissue. Practically, we label the interstitial fluid, the cerebrospinal liquor, with a fluorescent dye. We inject it either into the tissue, we inject it into, if it's in vivo, we inject it into one of the ventricles, the lateral ventricle, from where it spreads rapidly throughout the brain, and provides then sufficient contrast to see the cells in an inverted way. And because when the images are then acquired with a STED microscope and a very nicely aligned and 3D STED microscope, we have the chance to actually reach the anatomical ground truth. We can really resolve all the structures, the anatomical structures, mind you, not the molecular structures. We are far from the molecular ground truth, but in terms of anatomy, we can see all that there is. Yeah, there's not. So if we don't see something that's meaningful, that means it's not there. It's not just that it's not labeled; it's really not there. Yeah, and that gives us a new access into the complexity of the system. And as we can do this in a live setting, we can see the dynamics. And then so it's complexity, nanoscale, dynamics, and that's very cool. Yeah.